So, Yamada-san, thank you very much for your kind introduction. And it's a great pleasure and honor to be a speaker of this Microbiome Virtual International Forum. So thank you very much for inviting me. So here's my COI to disclose. And so we have been working on the gut microbiota, and the normal gut microbiota comprises about 1,000 bacterial species with more than 500,000 bacterial genes. So we substantially rely on the function of the gut microbiota. So therefore, the less diverse and less functional gut microbiota are often associated with several diseases, including immuno-related disease, metabolic disease, cardiovascular, and also cancer and infectious disease. So, and currently, the most effective way to manipulate and improve the function of the gut microbiota is fecal microbiota transplantation from healthy individuals. However, FMT has several limitations. FMT may fecal microbiota may contain pathogens, so therefore may be unsafe. Moreover, FMT is uncontrollable, and the effect is kind of by chance. So therefore, FMT should be supplanted with rationally designed microbial therapeutics consisting of well-characterized bacterial strains. So to identify effector microbes from the hundreds of members of the gut microbiota, we often take a reductionist approach. So in this approach, we first focus on one phenotype, such as specific immunocell subsets, such as the regulatory T cells, and we then try to narrow down the bacterial community while maintaining the focused phenotype, and to obtain a functionally relevant minimal effector bacterial consortium. So to do so, we usually take germ-free mice housed in sterile vinyl isolator like this, and we already introduced germ-free mice with usually human fecal microbiota for the purpose of reverse translation, and examine the phenotype. And if we see the phenotype such as regulatory T cell induction, we select the mouse and collect the fecal or cecal contents, and we transfer this microbiota to another germ-free mice. And then we introduce some perturbations to reduce the bacterial community and select a mouse that shows the strongest phenotype, such as regulatory T cell induction, and collect the fecal or cecal materials from the selected mouse and anaerobically culture them and isolate bacterial strains as many as possible. And then we examine the effect of the mixture of bacterial isolate, and subsequently we try to narrow down the bacterial community to obtain a functionally relevant minimal effector bacterial consortium. So to facilitate this approach, we have made a substantial effort to develop our technique to culture anaerobic bacteria. So now we have acquired extensive expertise, and we can isolate 60 to 70% or maybe more of the members of the human gut microbiota. And moreover, we have prepared non-biotic animal rooms, and in total, nearly 200 vinyl isolators are available just for our laboratory. So by combining non-biotic technique together with anaerobic culturing and NGS-based sequencing technique, we have tried to dissect the complex microbiota and identify effector bacterial strains. So by using this composite method, we have identified multiple immunomodulatory bacterial consortia, such as regulatory T cell inducing strains, Th17 inducing, Th1 cell inducing, and CD8 positive T cell inducing strains. In particular, 17 regulatory T cell inducing strains are currently under evaluation in a phase 2 clinical study as a part of Pfizer Breakthrough Growth Initiative uh, to treat ulcerative colitis patient. And the 11 seeded positive T cell inducing strains are in the middle of phase 1-2 study aiming at treating PD-1 antibody refractory cancer patients by Vedanta Bioscience and Bristol Myers Squibb. And recently, we have extended our research into the fields of pathogen infection, metabolism, and cancer. And today, among these new projects, I would like to introduce two studies related to pathogen infection. The first one is about the identification of trypsin degrading Paraplephotera species. So this study has been conducted by a very talented graduate student, H. Watanabe, with a brilliant former postdoc, Yokshan Lee. So we first performed the proteomic assessment of germ-free and SPF mice and found that several host protein levels were increased in the feces from germ-free mice in comparison to those from SPF mice. And among these proteins, this trypsin attracted our attention. The proteomic data was validated by trypsin activity test and Western blotting of feces and immunostainings of colon sections with an anti-trypsin antibody. The increased level of trypsin in the feces and in the colon of germ-free mice was obvious as compared to SPF mice. So we then examined the trypsin activity in the lumen of the jejunum, ileum, cecum, and colon of SPF mice. And this analysis revealed that the trypsin activity was very high in the small intestine, but the level suddenly dropped in the cecum of SPF mice. In contrast, in germ-free mice, the level remained very high in the cecum and the colon. So therefore, these results suggest that trypsin produced by pancreas is usually broken down by commensal bacteria colonizing the cecum. So therefore, we wanted to identify a bacteria that can break down trypsin. So to identify trypsin degrading bacteria, we obtained stool samples from 6 healthy volunteers, and the stool samples were each orally administered to germ-free mice, and fecal trypsin activity was examined. And the tested human fecal samples showed variability in the capacity to reduce the fecal trypsin activity in the recipient mice. For example, the sample from donor B did not reduce the fecal trypsin activity at all, whereas fecal microbiota from donor C, D, E, F all greatly reduced it. So among these effective microbiota colonized mice, we selected donor C microbiota colonized mice for the follow-up analysis. And actually, mouse C number 5 was selected, and the fecal content from this mouse were collected. And inoculated, the fecal contents were inoculated into whole groups of germ-free mice, and the mice were then treated with different antibiotics via the drinking water to perturb the microbiota. And then we carried the fecal trypsin activity in each group over time. So please look at this black line here. In mice colonized with the C number 5 microbiota without antibiotics treatment, fecal trypsin activity was nicely decreased by day 5. Interestingly, as shown in the red line here, fecal trypsin activity was further reduced by ampicillin treatment. In contrast, treatment with the other antibiotics abrogated this trypsin reduction effect. So therefore, responsible bacteria were enriched in these mice, but reduced in these mice. So therefore, we selected one of the ampicillin-treated mice, and we collected the fecal content from this mouse and cultured them in vitro and successfully isolated 35 individual strains. And we then examined the effect of 35 isolated bacterial strains by generating notobiotic mice. And we found that fecal trypsin activity and concentration were markedly decreased in 35 mixture colonized mice. So 35 mix is sufficient to degrade trypsin. So I don't go into detail, but using notobiotic pipeline, we successfully narrowed down from 35 to 14 to 9 and eventually to 3 bacteroides strains without compromising trypsin reducing activity in vivo. And we then performed an in vitro experiment using recombinant trypsin. And as you can see here, among the 3 bacteroid strains, this Paraplevotella clara strain, only this strain was able to degrade the recombinant trypsin. So therefore, we concluded that this Paraplevotella clara was responsible for the observed trypsin degradation. So then, to examine the mechanisms underlying P. clara-mediated trypsin degradation, we tested the effect of several protease inhibitors. And among inhibitors, as you can see here, the trypsin-specific inhibitor TLCK completely blocked P. clara-mediated trypsin degradation. So therefore, trypsin degradation by P. clara is likely mediated by trypsin itself, dependent trypsin autolysis. So, and interestingly, when P. clara was incubated with fluorescently labeled trypsin, in the presence of trypsin inhibitor TLCK, we observed the trypsin accumulation on the surface of P. clara within minutes. So therefore, P. clara probably expresses trypsin binding surface molecules, which facilitate accumulation of trypsin on the surface of P. clara and promote the trypsin autolysis. So to identify responsible molecules, we again used several inhibitors. And luckily, we found that both trypsin accumulation and trypsin degradation was severely blocked by tunicamycin treatment. So tunicamycin is known to inhibit the biosynthesis of LPS polysaccharide via interfering with bacterial VK activity. So therefore, polysaccharide binding surface molecules may be involved in the trypsin accumulation and degradation. And moreover, in whole genome sequencing of P. clara, we identified genes predicted to be components of the type 9 secretion system. So type 9 secretion system is known to be a transporting system that delivers the bacterial proteins across the outer membrane of bacteria and conjugates the protein with LPS polysaccharides, thereby anchoring the protein to the surface of the bacteria. So we hypothesized that the surface molecules delivered by type 9 secretion system might be involved in the trypsin accumulation and degradation. So to test this hypothesis, we generated POLU deficient P. clara strain. So POLU is an essential component of type 9 secretion system. So, and as you can see here, the polyu mutant P. clara showed a complete defect in trypsin degradation. So therefore, type line secretion system dependent surface molecules are likely involved in the trypsin degradation. So to search for such type 9 secretion system dependent surface molecules, we conducted proteomic assessment of the culture supernatants of P. clara in the presence or absence of tunicamycin. So we searched for molecules that were shed from the surface and accumulated in the supernatant in the presence of tunicamycin. And please look at the red bars here. Several bacterial molecules were found to be increased in the culture supernatants of P. clara in the presence of tunicamycin. So we then generated a series of mutant P. clara strains, disrupting each of those genes from left to right and examined all of them, although this was a painful and laborious experiment. But luckily, we found that the disruptions of 00502 gene or 509 gene resulted in abrogation of trypsin degradation, much like polyu mutation or WEK mutation. So although we don't know the structure 00502 or 509, perhaps delivered by type 9 secretion system, it binds to LPS and cooperatively recruit trypsin and promote its autolysis. So we are now evaluating this model. And interestingly, in addition to trypsin, P. clara appears to promote the degradation of another serine protease, TMPRSS2, in the intestine. So TMPRSS2 is known to be involved in the cleavage of spike protein and promotes the entry of coronavirus. So therefore, by reducing the levels of both trypsin and TMPRSS2 in the intestine, P. clara might inhibit the coronavirus infection from the intestine. So to test this hypothesis, we used murine hepatitis virus, which is a mouse coronavirus, mouse version of coronavirus. So since P. clara alone cannot colonize the mouse intestine, we colonized germ-free mice with two trypsin non-degrading mix together with wild type or 00502 mutant P. clara. And then the mice were administered with murine hepatitis virus via the intragastric gavage. And the difference was stunning, and wild type P. clara colonization contributed to the protection of the mice from lethal infection by this murine hepatitis virus, while 00502 mutant P. clara colonized mice succumbed to the infection. And the liver section from the 502 mutant colonized mice showed a lot of necrotic lesions, whereas wild-type P. clara colonized mice showed no or only a few, if any, lesions. So these data suggest that the trypsin-degrading P. clara colonization provides protective benefits to the host during the coronavirus infection, and may be also protective against SARS-CoV-2 virus infection in humans. So we are currently investigating this possibility using human samples or using mouse-adapted human SARS-CoV-2 viruses. So the second story is about the identification of unique bile acid metabolizing bacteria from the centenarians microbiota. So this study has been conducted by a wonderful staff scientist, Yuko Sato and Koji Atarashi, with the help of Damian Frichter from the Broad Institute. So in this study, we took an approach which is something that we call the bottom-up approach. So in contrast to or complementary to the top-down notobiotic approach, starting from fecal samples, fecal transplantation into germ-free mice, and narrow down the bacterial community to identify functionally relevant bacterial consortia. In this bottom-up approach, we started with small molecules identified via metabolomic analysis and sought to understand the response to bacterial species and then investigated their physiological relevance using mice. So we collected centenarians to samples. The centenarians aged 100 years and older are less susceptible to age-related diseases, including cancer and obesity and hypertension and cardiovascular disease. And moreover, they survived periods of hunger and several bouts with infectious diseases. So we reasoned that they might have beneficial microbes in the intestine. So in collaboration with Dr. Hirose of the Center for Supercentenarian Medical Research at Keio University, we aim to identify symbiotic beneficial bacteria from centenarians' microbiota that might contribute to the resistance to pathogen infection and other environmental stresses. So we recruited 160 centenarians, all Japanese, and collected their stool samples. And as controls, we collected feces from elderlies aged around 85 and young controls aged around 30. And we compared fecal microbiome and metabolites. And here is the metagenomic sequencing data, which was obtained in collaboration with Damian Prichter and Raunik Xavier at Broad Institute. And the microbiome composition of centenarians were found to be different from those of the other groups. And centenarians showed increases in microbia such as Akkermansia and proteobacteria such as Desulfovibrio species. And there was also a decrease in actinobacteria in centenarians as compared to controls. And a more interesting observation was made by the analysis of fecal metabolites, particularly fecal bile acids. And as you can see, fecal bile acid compositions of the centenarians were pretty different from those of the controls. In particular, this bile acid, isolytocolic acid, LCA, was specifically enriched in the centenarians' stool samples. So this is the Keck secondary bile acid biosynthesis pathway depicting transformation from the primary bile acid canodeoxycholic acid CDCA to the secondary bile acid lytocolic acid LCA. So this transformation, which involves multiple enzymes, should be conducted by the microbiota. And importantly, the generation pathway for isoallo-LCA had not been reported. So we predicted that isoallo-LCA might be generated from this 3-oxo delta-4-LCA via 3-oxo allo-LCA by 5-alpha reductase and 3-beta hydroxysteroid dehydrogenase, 3-beta HSDH. So this was because isoallo-LCA is structurally similar to 3-beta androstenediol, and 3-oxo allo-LCA is structurally similar to dihydrotestosterone, and 3-oxo delta-4-LCA is structurally similar to testosterone. So N5AR is known to mediate the conversion of testosterone into dihydrotestosterone, and 3-beta HSDH mediates the conversion of dihydrotestosterone into 3-beta androstenediol. So therefore we predicted that 3-oxo allo-LCA might arise from the 3-oxo delta-4-LCA via pathway similar to the 5-alpha reduction of testosterone by adding the hydrogen atom to the carbon 5 position after the cleavage of the double bond between carbons 4 and 5. So we also predicted that the subsequent transformation from the 3-oxo-RLCA to iso-RLCA might involve 3-beta-HSDH, mirroring the 3-beta-HS mediated conversion of dihydrotestosterone into 3-beta-androstenediol by replacing the 3-beta by replacing the oxo group into hydroxy group at the carbon 3 position. So anyway, to identify iso-RLCA generating bacterial strains, we selected this person, C91, because this individual showed a high level of iso-RLCA in the feces and maintained a relatively good health condition. So from the stool sample of C91 centenaria, we isolated 68 bacterial strains. So here is the list of the 68 isolated bacterial strains, which contained pretty diverse strains, species. So we then tested their bile acid metabolizing capabilities using in vitro culture system. So we used CDCA or 3-oxo-delta-4-LCA or lithocolic acid as a substrate, and each individual bacterial isolate was cultured in vitro with either of these bile acid compounds. And the culture supernatant were analyzed by LCMS. And as you can see, here, incubation with CDCA did not result in accumulation of iso-LCA in any of the cultures of 68 bacterial strains at all. And again, when cultured with LCA, none of them was able to generate iso-LCA at all. But excitingly, when we used 3-oxo-delta-4-LCA as a substrate, high levels of iso-LCA were detected in the cultures of several bacterial strains. So here is a little bit zoom up of this part. So among the 68 bacterial strains tested, strain 3 Parabacteroides meldae and strain 19 Odoribacter laneus and strains 21 to 24 undefined Odoribacteriaceae strains were found to be able to transform from 3-oxo-delta-4-LCA to iso-RLCA very effectively. So we then sequenced the genomes of these strains and found that P. meldae strain and Odoribacter strains carry gene clusters containing putative 5-alpha reductase and 3-beta HSDH genes. So we thus generated 5-AL gene or 3-beta HSDH gene deficient P. meldae strains by conjugation-mediated plasmid transfection and homologous recombination. So when 3-oxo-delta-4-LCA was used as a substrate, wild-type P. meldae was able to produce iso-RLCA in vitro very effectively, whereas 5-AL mutant P. meldae strains, actually we obtained 3 mutant strains, all failed to produce iso-RLCA. But in contrast, when 3-oxo-RLCA, so this one, was used as a substrate, both wild-type and 5-AL mutant strains were able to produce iso-RLCA effectively. Moreover, the 3-beta HSDH deficient strains failed to generate iso-RLCA when it was incubated with 3-oxo-RLCA. So these results all supporting for our predicted transformation pathway from 3-oxo-delta-4-LCA 2,3 oxo RLCA and 2 iso-RLCA by 5-alpha reductase and 3-beta HSDA. So now, Eric Paymer at U Chicago and Casey Elliott at North Carolina State University and several other groups have shown that secondary viruses such as LCA and DCA can inhibit the growth of antibiotic-resistant Clostridioides difficile. So therefore, we tested the effect of iso-LCA on C. difficile, and we incubated C. difficile with different concentrations of iso-LCA or other virus compound and measured the growth of the C. difficile over time. And surprisingly, iso-LCA showed a very strong effect. And even at the lowest concentration tested in this experiment, C. difficile did not grow at all in the presence of iso-LCA. And actually, MIC19 was quite low as compared to the other viruses tested. And the scanning electron microscopy revealed that iso-LCA was very much bactericidal and disrupted the cell wall of C. difficile. And interestingly, besides C. difficile, iso-LCA was also inhibitory to vancomycin-resistant Enterococcus faecium. So iso-LCA MIC19 against VRE was around 2 micromolar, which was much lower than that of the other viruses tested. And scanning electron microscopy of VRE revealed that iso-LCA produced morphologic alterations such as large and explosive bacterial cells. But in contrast, E. coli was totally insensitive to iso-LCA and also as a virus tested. So actually, we have tested several gram-positive and negative pathogens and found that to all the gram-positives, including C. difficile, VRE, and Staphylococcus aureus, iso-LCA was very effective and exerted a very strong growth inhibitory effect. But in contrast, gram-negative pathogens such as Klebsiella, E. coli, Salmonella were all insensitive to all the viruses tested, including iso-LCA. So therefore, iso-LCA producing bacteria such as Odoribacter and Parabacteroides meldae are likely contributing to prevention of gram-positive pathogen colonization, which might be contributing to the maintenance of intestinal homeostasis in centenarian. So we are currently testing this possibility. I mean, the contribution of iso-LCA producing microbes to the longevity, healthy longevity. So these works has been conducted with kind help from many collaborators, including Romnick Javier, Dan Littman, Michael Fishbach, and many others. So I stop here and thank you very much for your attention.